Hello and welcome also from my side to today's webinar. My name is Eric Lutsch. I'm an application scientist with Tecan, specifically for imaging. My background is in cell biology, more precisely in regenerative physiology and aging. And I will be your host for today, guiding you through today's webinar. Please make sure to ask questions throughout the session, as we will have a Q&A session in the end. Please just type your questions in the chat and I will address all your questions in the end of the session. Tecan's global operations. I briefly want to introduce Tecan and its different sites across the globe. Here at Tecan, we develop and produce state-of-the-art laboratory automation devices. Highlighted in green is the site here in Austria, where we are the provider of the detection and washer instruments and have almost 40 years of experience in the analytic instrument market. Our mission at Tecan is to improve people's lives and health by empowering our customers to scale healthcare innovation globally. Tecan does this from research to the clinic by enabling the customers to further understand disease, help increase effective medicine in regard to supporting drug development and discovery, support drug production in higher quality and scale, and enable for personalized medicine and diagnostics, which therefore give means for more precise therapy approaches and help managing disease risks. Our main customers range from academia and research centers over biotech and pharma, genetic testing and reference labs or clinics. And our main applications reside in the field of genomics, cell and tissue culture, proteomics, or medical mechatronics. Bridging the gap to in vivo relevance. A common starting point from molecular and cell biology-based research is 2D cell culture using cell lines. For more than a century, it has been the gold standard and a very powerful method to help scientists answer biological questions due to the high experimental flexibility, but also the reproducibility and ease of handling. However, while making many groundbreaking discoveries, there are also significant drawbacks, as findings in 2D often fail to replicate and translate to what happens in the body. As you can see, there is a large gap between the 2D models where cells are grown on flat and rigid surfaces, with all cells exposed to the same environmental signals, and animal or human models. To bridge this large gap, we need to move towards 3D cell culture. Cells grown in 3D, such as spheroids and organoids, allow for greater interactions not only in between cells, but also with the environment and extracellular matrix, where they experience a gradient formation, meaning nutrients and oxygen, but also drugs penetrate to different degrees, particularly of importance when considering tumor response. Given the right environment and signals, cells have the potential to differentiate into various cell types and form structures to mimic organs or tissues in vivo. By adding these 3D cell models into experimental workflows allows us to bridge this gap and give more meaningful insights and physiologically relevant information. As organoid models well retain the morphological phenotypes, cell functions, biological behaviors, and genomic profiles of parental tissues, the key reasons for transitioning from 2D to 3D cell culture are the higher biological relevance, better quality of assay results, and enhanced cell viability and long-term cell culture with higher yields. So what are the main differences between these models? As you can see, spheroids are simple 3D in vitro structures generated from single cell type. This comes with a gentle learning curve with regards to handling of cells and matrix. We can add different cell types which are found in the microenvironment or matrix as another layer of complexity. As this brings us a bit closer to the in vivo situation, these models still lack differentiation and polarization cues, in vivo gene expression, and drug responses. So we do not see the types of structures we do see in vivo. However, these deficiencies are addressed by organoids, which are complex three-dimensional structures derived from stem cells, capable of self-organizing, self-renewing, and differentiating into multiple cell lineages. As they are very complex to cultivate, they require expert handling and have a much steeper learning curve. The benefits are that gene expression profiles are maintained, and then they can recapitulate the in vivo environment and drug response. These models can mimic the organs which they originate from and recapitulate to some degree the functions of these organs. Patient-derived organoids hold unique promise for personalized medicine, as they offer promising opportunities to model tissue development, disease, and drug response in vitro. The need to bridge this gap of physiological relevance can be clearly demonstrated by looking at the drug discovery funnel. Using the traditional methods as cell-based assays and 2D culture screens are heavily utilized. With over 10,000 candidate drugs entering the funnel initially, the discovery has a huge attrition rate over the 10+ years of the drug discovery process and massive cost to R&D, which produces just one successful drug. With less than 10% of the anti-cancer drugs that enter clinical trials reach the market. This high failure rate can be linked to inefficient preclinical models with low predictive power that simply cannot recapitulate the in vivo situation. However, organoids have demonstrated to accurately predict patient response to targeted therapies in the case of colorectal cancer, lung cancer, or breast cancer, just to name a few. A major cause of drug withdrawal from the market is drug-induced toxicity. In this case, liver organoids have proven to play a vital role in predicting drug-induced liver injuries. These results have also led to a change in legislation as the FDA Modernization Act was signed into law in December 2022, which aims to reduce reliance on animal testing in drug development by allowing the use of alternative testing methods such as organoids or organoid chips in preclinical drug research. Highlighting the key applications of organoids were, for example, investigations of cellular interactions with the surrounding microenvironment and other cells in culture, or cell differentiation and proliferation, the evaluation of substance toxicity and sensitivity to potential drugs are major fields of organoid application. The evaluation of the regenerative potential, gaining insights into complex gene profiles, but also the diagnostic and therapeutic potential of organoids is of high interest to scientists. For individual patients, organoids have already been used to identify beneficial treatments, so providing true personalized medicine. They may further serve as autologous material for cell replacement therapies or even future organ transplants, with the possibility to correct disease-causing mutations by CRISPR-Cas9, for example, before transplantation. Organoids grown from groups of patients can be used to model disease, for example in infection biology, but also to understand specific genetic mutations. For drug development, organoid biobanks are a very promising tool for drug discovery. Further, Kidney and liver organoids in particular may help in the future to complement or even replace current animal tests for toxicity. Both adult stem cells and pluripotent stem cells derived organoids can be used for the areas of biomedicine. To date, we're facing key challenges and various limitations of organoid cultures. So the high variability, where standardization and reproducibility regarding consumables, so to say the batch-to-batch variability, where differences in extracellular matrix and growth factors can lead to inconsistent results. But also handling of the matrix or growth factors, as well as the cell culture handling itself, are major drawbacks to organoid cultures. Also protocol differences, so the lack of standardized protocols across labs, make it hard to compare results. The complexity of the microenvironment, so limited immune cell systems or supporting stromal cells are issues. The mimicry of in vivo conditions, so recapitulating the complex cellular interactions, gradient formation, and mechanical forces found in real tissues is very difficult to achieve. The limited vascularization, as most organoids often lack blood vessels completely, limiting nutrient and oxygen delivery, especially in larger organoids. Also the analysis and imaging, where we see difficulties in imaging as the 3D structures are very large and are more difficult to image and analyze compared to 2D cultures. The sample preparation, where sectioning and staining of organoids for analysis can be very challenging. Scalability and throughput, as the cultivation of organoids is very labor intensive. Manual handling and monitoring are very time consuming, which also leads to automation challenges, where adapting organoid culture to high throughput platforms is technically challenging and very demanding. Also the genetic and phenotypic stability, especially in long-term cultures where the genetic drift plays a vital role, where organoids can accumulate mutations over time, affecting their reliability. Secondly, the phenotypic heterogeneity, so the variability in cell types and the organization within and between organoids make it difficult to compare the results. Next, I want to highlight our detection instrument portfolio here at Tecan, where we offer comprehensive reader and washer solutions. Highlighting on the top specifically for the organoid purpose is the Spark Cyto, which is our high-end multimode reader with imaging capabilities and corresponding instrument control software, namely the Spark Controller, where you can perform method definition, but also data acquisition and analysis in the image analyzer software. Our flagship multimode reader in detection is the Spark Cyto, where imaging is combined with the best-in-class multimode reader. So it's a powerful tool for multiplexed analysis and to get the most data out of your experiment. The Spark Cyto offers effortless monitoring of organoid growth, morphology, and treatment effects in real time. The system has integrated incubation and parallel data acquisition features, streamlining experimentation, while AI-driven imaging algorithms ensure accurate identification and analysis of organoid structures. Designed for the ease of use, the system automates entire workflows, from imaging to data analysis, making it ideal for researchers new to organoid studies. Highlighted here in blue is our absorbance with high-speed monochromators, so very fast in recording of absorbance spectra, but also for nucleic acid quantification in low volumes when combined with our nanoquant plate. We offer fluorescence with fusion optics, meaning a double monochromator with filter optics to enable very flexible but also highly sensitive fluorescence readouts. We have fluorescence top and bottom, time-resolved fluorescence, TR-FRET, or fluorescence polarization. We also support luminescence, so supporting glow and flash lumi, multicolor lumi, and spectral recording of lumi. We have imaging capabilities for live cell monitoring in bright field and fluorescence, and offer environmental control and real-time experimental control. So we have an integrated automated lid lifter system. We have a cell incubation chamber where we can regulate temperature and gas, and counterfeit evaporation via our humidity cassette. Additionally, we have a very intuitive and user-friendly software, namely the Spark Control. The Spark Cyto is available in 5 different configurations. We have 3 different objectives, a 2x, a 4x, and a 10x optical magnification combined with performant digital zoom. Adding a new dimension of image analysis with the new Spark Cyto 3D AI, where we can automatically monitor 3D cell cultures, including the analysis of spheroids and organoids in real time using an AI-based analysis. We have real-time data display on live cell experiments, giving you real-time feedback while doing the experiment. We have a high flexibility and scalability with various supported plate formats and offer whole well imaging for 96 and 384 well plates. We can also multiplex, as already mentioned, our assays, for example, with cell titer glow or other luminescent, fluorescence, or absorbance-based assays. And we offer label-free and fluorescence-based and fluorescence AI-based segmentation. The instrument is also capable of automated set stacking and the creation of total projection images, targeting drug discovery, cancer research, and personalized medicine, and helping to rapidly translate lab results into meaningful insights. Highlighting a few capabilities of the Spark Cyto 3D AI, in this case for single spheroid tracking. So you can do segmentation of single spheroids in bright field and fluorescence channels, and you can do also spheroid growth monitoring under different conditions. So no matter if you're investigating different seeding densities and the development of your spheroids, highlighted in those 2 fields down here, or if you want to analyze different concentrations of drug treatment over time, this is possible with the Spark Cyto, as it gives you real-time feedback while your experiment is taking place. So in the case of multiple objects in a well, this can also be done by the Spark Cyto 3D AI. As you can see, the segmentation and quantification of multiple spheroids in this section here on the left, they have been cultivated in matrix. You can also perform the creation of set layer via the automated total projection images. And this is very useful when having multiple objects that reside on different set heights. As you can see in these images below, this is a single planes image, so only one set layer where clearly some specimen are out of focus and not being analyzed, whereas in the total projection of the same image, all of the objects are in focus and all of the objects are also being analyzed. So this enables to capture and analyze all objects well faster by automated set stacking and analysis, increased data range and reproducibility, and less storage space through projected images. For further automation of this workflow, Tecan offers the Fluent Automation Workstation. It's a one Tecan solution for end-to-end workflow automation. The Fluent prevents cell culture contamination with the integrated HEPA hood and biosafety cabinet. It enables for optimal cell viability and minimize assay variation with integrated automated incubators with environmental control. We can monitor cellular health and benefit from multiplexed analysis with the Spark Cytos imaging capabilities, whereas the Fluent finger exchange system enables handling of both tubes and microplates with a robotic gripper arm. To automate your routine cell culture work, such as cell seeding, medium exchange, and cell harvesting on Tecan liquid handling platforms. It is integratable with centrifuge, incubators, or cell imaging systems with the Tecan LabWorks to automate that complex workflow. Another feature in this regard is the Dream Cell. It's a variation of this system. It's a modular platform for automated cell culture in a sterile environment, adaptable to customer needs. It offers rapid aspiration and dispensing with the CultureBot to streamline handling of large liquid volumes. The CultureBot is a multifunctional module enabling large volume aspiration and dispensing with a built-in tilting and knocking functionality for routine cell culture maintenance steps, like medium exchange and cell passaging. It offers a greater cell culture standardization by reducing hands-on time and human error. And it offers to run multiple plates at a time by scheduling liquid handling, incubation, and cell growth monitoring tasks. I here want to mention the Duo digital dispenser, which is not only able to handle liquids, but also different cell types for routine cell culture. So I want to summarize our key takeaways for the Spark Cyto 3D AI. We can automatically monitor 3D cell cultures in real time with bright field and fluorescent-based segmentation with 3D AI algorithms. It offers kinetic measurements in a continuous fashion or open monitoring, giving you high experimental flexibility without blocking the system. It is able to multiplex with cell titer glow, for example, or other luminescence readouts, or fluorescence and absorbance-based assays. It offers the options for injectors, so also in the standalone Spark Cyto, we can do, to a certain degree, liquid handling, either for your assay development or routine cell culture work. It offers the lid lifter and the real-time environmental control system. This leads to translating lab results into meaningful insights. And this is done with a very intuitive software with a smooth learning curve. Targeting drug discovery, cancer research, or personalized medicine, automating your workflow with the Fluent Workstation and the Duo digital dispenser. And we also offer strong pre and post sales support and instrument simplicity. So whenever you are struggling with your assays, we are always willing to help you either remotely via the help desk or expert line, or directly through our sales colleagues. I just want to give you a couple of examples of applications we already are doing. So this is drug response in patient-derived organoids done by Hub Organoids, where they assessed the fluorouracil and staurosporine-induced cell death in colorectal cancer patient-derived organoid models. The quantification of drug-induced cell death via cyquant proliferation assay. So in the images below, you can see the colorectal cancer patient-derived organoids 5 days after incubation, stained with the cyquant dye and Nunc blue in a 96 well plate on the left side, imaged with the 2x objective. So this is a whole well image. And on the right side, this is a 384 well plate, imaged with the 4x objective, also giving you a whole well image without the need for tiling or stitching. And on the right side, we can see the percent viability that was assessed for the 5FU treatment. So the colorectal cancer PDOs after 5 days of exposure to the treatment at different doses, again in the 96 well plate on the top and the 384 well plate on the bottom. One of our most recent applications is the functional ADME tox testing in early stage drug development, done in collaboration with Mimetas and their organoready colon organoid model, where they were assessing drug absorption, distribution, metabolism, and excretion and toxicity in early stage drug development. So as the gut epithelium plays a key role in modulating systemic drug exposure and local toxicity through active transport and metabolism, traditional in vitro models often lack the physiological complexity of the human intestinal tissue, limiting their use in assessing transporter and enzyme function. Compatibility with the Spark Cyto enables high throughput, non-destructive, and multiparametric assessment of transporter and enzyme activity. So the integration of quantitative fluorescence and luminescence detection with real-time imaging provides a robust and scalable workflow, enhancing assay reproducibility and generating comprehensive multiparametric data from a single experimental setup. These results support the organoready colon organoid model as a reproducible, human-relevant platform for preclinical evaluation of intestinal toxicity and drug metabolism. So the BCRP encoded by the ABCG2 gene plays a key role in drug efflux and xenobiotic protection at physiological barriers in key organs. Mimetas used the Spark Cyto to monitor organoid growth in real time by combining standardized protocols with advanced live cell imaging. Researchers can quantify key parameters like fragment size, morphology, and growth behavior. This reduces the variability, provides donor-specific insights, and leads to more reproducible results. With these organoids, Metas produces ready-to-use assays for preclinical studies to accelerate drug discovery and help reduce animal testing. Just highlighting a few other applications. So I want to highlight a few other customers that are already working with organoids and using the Spark Cyto for their research, such as Dr. Christoph Deven from the University of Antwerp, where they're doing a high throughput format in a 384 well plate, using an automation protocol for real-time and endpoint assays in bright field and fluorescence imaging, plating organoids using the Spark Cyto injector, and treatment with the D300E digital dispenser. I want to make a statement here that the D300E is no longer available, but the Duo digital dispenser is available in this regard and with additional features. And they did the data analysis with their own analytical software. Another customer is Daniel Engel at the Salk Institute, where they're dealing with patient-derived organoid modeling of pancreatic cancer. In this regard, doing precision medicine and drug screening, also with the Duo, monitoring and analyzing drug response to those patient-derived organoids, comparing intracellular ATP to confluency. And this helps them to predict clinical outcomes, so patient treatment actually match organoid treatment. For Julia Krishna at Z Predictor, where they have this wonderful RBreast 3D cell culture model with cancer tumoroids, and they do a sequential live dead assay and cell titer glow analysis. So they test therapeutic agents, anti-cancer drugs in this regard, also with the D300E or Duo, and evaluate the response in real time using our imaging system with the Spark Cyto. They also do the live dead staining, so the calcein and etidium homodimer staining, and fluorescence imaging, and endpoint cell titer glow luminescence readouts. Finally, I want to highlight a few webinars and literature you can do reading up on. So the Mimetas already mentioned, our latest webinar and application note that was launched, but also highlighting different other application notes which are all found on our homepage, and also some very new featured Tecan journal advancing endometriosis research to improve outcomes for women worldwide, or improving organoid reproducibility through real-time imaging articles. Also, We have a variety of different webinars also found on our homepage. You can have a look at to get a better view on what some customers are already doing and get a better understanding if this is a suitable system also for your research. With this, I want to thank you a lot for your attention and very much looking forward to the Q&A session. And I hope I can answer all your questions you have. Yeah, feel free to ask anything. Thank you again and talk to you in a bit.